This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me today, I have Dr. David Orton of the University of York, professor of zooarchaeology. Good morning. How are you?
Good morning, Gabby. I'm good. It's afternoon here.
Right, rather good afternoon. Thank you so much for joining us. I think you are actually the first zooarchaeologist that we've had on the show, which for listeners, I know I'm saying zooarchaeologist, but it's zooarchaeologist. I don't really know why we pronounce it that way. Maybe they don't pronounce it that way in the UK. What do you guys say?
I don't think I noticed the difference there, to be honest. I say zooarchaeologist.
Yeah, in the US, at least with the zooarchaeologist that I worked with, Dr. Joanne Bowen, who was at Colonial Williamsburg, she always said zooarchaeologist, so that's how I've always pronounced it. But now that I look at the word, it definitely is zooarchaeologist. Well, that's okay. Just differences across the pond. Well, welcome. Can you describe what zooarchaeology is for listeners?
Okay, so zooarchaeology is the study of the long-term history of human interactions with animals. Now, overwhelmingly, that means we're looking at their physical remains. So it's always tempting to say we're people who look at animal bones, because that's mostly what we do. But we're looking at those remains specifically to tell ourselves more about human societies in the past. And that means that we can also draw on lots of other forms of evidence as well. So documentary sources, just like other archaeologists draw on history, we also do that, but also all the same kinds of molecular analyses that people apply to human remains, we can apply the same kinds of things to animals as well.
Yeah, that was a really great summation. Zooarchaeology, I'm just going to do that this entire show, I think, is really cool. I got the opportunity, like I mentioned, I had a professor when I was in college who was the zooarchaeologist for Colonial Williamsburg, which is a site in Virginia. I saw that you did a paper called The Ratting of America, and there were quite a few sites that took bones from sites that I had actually also worked on, which is pretty cool.
Yeah, I have to say that was a paper I came onto late and helped with some of the analysis and the visualization and so on. But it's all credit to Eric Geary in Trent for sourcing material in the first place.
Yeah, I will say it was a really great paper, and it was just kind of nice to see like a little callback to my earlier days. But no, one of the really cool things about zooarchaeology is, like, in order to do your job, you are sitting there. I know you said it's not just bones, but like you are sitting there with quite a few bones, and you go through them and you figure out this belongs to X animal, this belongs to Y animal. Can you kind of describe what a zooarchaeologist's kind of typical day at a site might look like?
Oh, okay. Well, so it's a long time since I was working in the field myself. I've ended up being the kind of person who is more more synthesizing data, visiting archives, collecting individual samples to do analyses on, and so on.
That's another typical day, to be honest.
Yeah. So I guess for me, a typical day, leaving aside teaching and that kind of thing, would be sending a lot of emails to people, asking about specimens, asking about old reports, and do they know where the bones are now, and that kind of thing, updating people on results that have come through, working with databases and spreadsheets. It's not glamorous, but it's a big chunk of what we do. And writing when I can get to it. But yeah, when we are working in the field, and it's in an ideal world, the people working on the bones are going to be out there in the field working alongside the field archaeologists. Doesn't happen as often as we'd hope. So big research projects, big university-funded projects, that can be the norm. More often, people are drafted in after the fact. So whether that's in a CRM context or whether that's a research project that wasn't quite well enough funded to have somebody on site the whole time.
Mm-hmm.
So a lot of the time, we're trying to kind of contribute to the archaeology of the site, but doing the whole thing somewhat retrospectively, which can be a challenge. So it's a great privilege when you do get to be out there in the site. And I worked for a long time at Çatalhöyük, the famous Neolithic site in Turkey, where we well, the whole project made a point of having all the specialists on site. That was the director Ian Hodder's kind of concept from the outset. And that was fantastic because although I rarely got into the trenches and got my hands dirty, every day you would be getting kind of immediate feedback from the field archaeologists about what they were finding, and you could give, well, not immediate because it takes a bit of time to actually look through the bones, but within a few days you could be feeding back on the kinds of things we'd found.
Yeah, no, it's really cool. I think like to this day, one of the most impressive things that I've seen was Dr. Bowen going through a collection of bones and just like looking at like tiny carpals or whatever and being like, oh, this is clearly Suscrofa or the pig. This is clearly a cow. This is like a juvenile raccoon. This is probably a squirrel. It's amazing like how fast you guys are at that. I can't believe it, honestly.
Yeah, you can be. You get very used to the species that turn up in the sites you work on. So it depends where you are in the world. We have it easy here in Western Europe. There's not that many species typically. As you get either further south into tropical areas or, for that matter, further into Eastern Europe and into Asia, say, there are way more species. North America has more species than us too, to be honest.
I will say,
if you move to a new area, there's always a steep learning curve, learning the local fauna.
yeah. One of the interesting things that I worked on for my undergraduate thesis was it was one of the things that zooarchaeologists often do is they help with analysis of dietary patterns in areas because people eat animals. It's really cool because you just never know what you're going to find. It kind of depends on the assemblage. And for listeners who are new to archaeology, an assemblage is just like a grouping of objects, or in the case of bioarchaeologists, bones from a particular site. But you never know what you're going to find in an assemblage, and especially in places where there is a lot of trade. So, sorry to keep going back to Colonial Williamsburg, but that's where most of my experience is. But we did have quite a few kind of exotic species because it was a mark of high status to eat interesting animals. So like every once in a while we'd have to pull out a book and just like flip through it until we found something that matched the bones that we had. It's just kind of like very out there things brought up from the Caribbean, things brought all the way from Africa. It's really cool.
Yeah, it always makes life more interesting when you get something that's a bit of a challenge, something you weren't expecting.
Yeah.
I mean, yeah, you're right that 90% of what we do probably is about diet, 90% or more. And if you work on a historic period site, or if you work on most Neolithic or later prehistoric sites in Europe, a huge percentage of what we're looking at is cattle, sheep, goat, pig, some deer. It's a relatively small range of species over and over again. We get very, very good at doing that. It's always nice when something more surprising comes up. And I've kind of in the recent years of my career, I've more and more tried to work on things which aren't just about what percentage of this species did they eat, what percentage of that species did they eat, but particularly things to do with either the animals that live around humans, so particularly in urban contexts, but what you might call settlement fauna, and then also animals that are being traded. So again, it's about diet. We did a lot of work on fish and the development of the fish trade, which is, again, it's fundamentally about what people are eating, but it's not just, when do they start eating cod? It's also where are they sourcing that from? When do we start seeing long-distance trade coming in? What's the political context of that long-distance trade? And so on. Not to mention what impact that then has on fish stocks.
Yeah, there's an incredible nuance to your work, and it's really, there's quite a bit of breadth as well. Sometimes I definitely have those memories of like reading through zooarchaeological reports that are just like, it was just inventories of what animals they found in middens, which is it's the archaeological word for trash pits. Because that's where animal parts end up. And it's not sometimes not the most exciting thing. And that was one of the really cool things about the studies that you're working on, which actually this is a great transition. You are currently working on this really cool project called Ratus, which is about rats in Europe. Can you kind of explain the main thrust of the project? What are you guys trying to do?
Okay, so the starting point here is that rats are not from Europe originally. So there's two species of rats that have ever been in Europe, at least since the last ice age. Before that, all bets are off, I think. And those are the black rat and the brown rat. And they're both originally from Asia, different parts of Asia. But of course, they get to Europe at some point, and they get to Europe because they're able to take advantage of kind of an ecological niche around human settlements, because they end up living in close proximity to humans, eating human-derived food sources, whether that's raiding stored food or eating discarded refuse. They're really able to thrive in that context, which then gives them the opportunity to hitch a ride on land transport, on ships, and so on. And at some point, they then spread into Europe, later on into, perhaps earlier actually in some cases, into Africa, later on into the Americas via colonial period shipping and so on. But there's not that good an understanding of the timing of all those processes. And it's not just about the initial spread, it's also about having the right settlement conditions to support rats. So one of our main aims then is to trace when these two species are first of all reaching Europe. In the case of the black rat, it's at some point prior to the Roman period. It's really not that clearly defined at the moment, but I would guess probably somewhere in the 1,000 years or so before the Imperial Roman period. So the last millennium BC, let's say. But there are some earlier reports too that need to be checked out.
And are you using historical reports? Are you using physical remains?
Okay, so for the black rat spread, it's about physical remains, and we're not actually kind of pushing this back too early. We're focusing more on stuff a little bit later, which I'll come to in a moment. But it's mostly a matter of finding reports of early rats, going and checking them out, seeing if they really are rat bones. Most zooarchaeologists don't really work with the small stuff, so you do get misidentifications from time to time. We had one that was supposed to be, I think, about 1,800 BC from a site in Syria recently, which has been been stored in a collection in America for quite some time. And when we checked it out, it's not a rat at all, as it turns out. So yeah, it's that kind of thing.
What was it?
We're not 100% sure. Probably some kind of vole, but there's a lot of species of kind of slightly smaller than rat rodents in that part of the world.
Mm-hmm.
Make it very hard to identify unless you have cranial remains. And it was something that wasn't in our proteomic identification database, put it that way.
Okay.
If we check out a specimen and it does actually turn out to be a rat, then the next thing is to get a direct radiocarbon date on it. And this is something that 10, 15 years ago would have been a bit problematic, but the technology is always getting better for kind of minimizing sample sizes. So these days we can take a subsample even of a tiny rat bone and still get a radiocarbon date out of that. And then, of course, having established that we really do have early rat finds, we can consider whether it's worth including them in an archaeogenetic study, which for now we're focusing on later periods, but we're likely to roll back into that period as well. And when we're looking at this kind of classical or even earlier spread, we can't really use written sources. So for many of the times and places involved, there are no written sources. Where there are, so for example, when we have Greek or Latin sources available, they're not likely to allow us to pin down what species we're talking about. So I don't know the Greek so well, but in Latin, the word mus, or mures plural, basically applies to any small rodent. And you really, maybe based on context, a classicist or a historian might be able to get some idea of what kind of a rodent they're talking about. If it's causing a problem around a settlement, then it's quite likely to be either a rat or a mouse. But telling which one it is pretty much impossible. On the other hand, when we're looking at the spread of the brown rat, which is much more recent, we think they probably got to Europe in the 18th century, although there are some kind of suggestions it might have been a bit earlier. In that case, documentary sources are one of our main lines of evidence. By the 18th century, certainly by the middle of the 18th century, there's a huge kind of flourishing of natural history as a formal discipline. You have, obviously, Linnaeus's the definitive edition of Linnaeus's work published in 1758, and people are doing similar things for decades before that. But it really takes off in the second half of that century. And so we have lots and lots of reports where people are starting to try to be really specific about pinning down the species they're talking about, and also making a point of citing each other. So we don't quite have the formal citation practice at that point that we're used to these days. It can be a little bit tricky. People kind of copy stories from each other, and you realize it's been copied, but it's not clearly referred to where they got it from and that kind of thing. But when they're identifying the species, they'll always say kind of, whatever name they give to it, they'll make a list of all the other names that different sources have made to it as well. So we can really start tracing things back and working out who's talking about the same species, who's talking about something different. So that's a completely different world, really, to when we're looking in the Roman period, say.
Yeah. Linnaeus, for listeners, is the guy who came up with our kind of double-barreled taxonomic system, right? So like when you think of Homo sapiens with those two, he's kind of the guy who came up with that. There's like a whole larger system too. This is the thing that you remember in grade school.
He formalized
He
it a lot. To be
formalized
honest,
it. Yeah.
he did some very impressive work, but I think it's sometimes overstated a bit how much he changed what was already happening.
Yeah, I think that's totally fair. There was like this kind of culture of, like you said, scientific inquiry that was going around at the time.
Yeah, and it causes us some problems in a way because this is a tradition which is derived primarily from Northern Europe. And so when we're looking at, one possible route by which the brown rat might have spread into Europe is up through the Caucasus and southern Russia,
Mhm.
in which case coming via Iran, basically via Persia, around the back end of the Safavid Empire. But in the Persian traditions, people are writing about the environment, people are writing about animals, and I'm talking a little bit through my hat here. I don't know that much about the Persian tradition, but what you're not getting is it's not tied into the same terminology that we would have in the Northern European tradition. So we can't be sure of the same species. So when we look at what dates we have definitive mentions of the brown rat at in southern Russia, the Caucasus, and so on, the dates get later as you go south. And we would expect them to get earlier as you go south, but they get later as you go south because those dates are all coming from naturalists who are working for the Russian Academy in St. Petersburg, but
Mhm.
mostly actually German, and who are using that tradition. And as kind of this Russian-sponsored intellectual inquiry spreads south, we start to get data that we can slot into our systems. And one thing that I really hope somebody will do in the future is actually explore these kinds of issues through the Persian literature, which is something which is not accessible to most of us, really. But we have some quite big biases in the data driven by the fact that we're looking at a period that is just around the time people are starting to develop terminology. I don't want to make it sound like it's in any way superior to other approaches to it, but it's terminology which chimes with what we are using ourselves.
Right.
Yeah.
Yeah. One of the interesting things, I went and read up about black and brown rats before we started talking, and one of the things that struck me is that brown rats, the taxonomic name is Rattus norvegicus. I don't know if I'm pronouncing that right, but it's basically Norwegian rat. And there's just like a big question mark about like why why they're calling them Norwegian rats, which I think ties into a lot of the work you're doing about like where the heck did these come from? Like, why
Yeah,
did someone decide Norway?
I think we can be fairly confident that it's a misnomer.
Yeah.
I mean, we're fairly confident that Norway's not where they're reaching Western Europe from. Not least because the earliest mentions we have in Norway are something like 1762. Yeah, 1762. Whereas we've got people using the phrase Norway rat, so not the Latin, but the vernacular English phrase Norway rat in newspapers in Dublin in, I think it is 1729 already. So whether they're referring to the same species or not, we can't be entirely sure. It seems likely they are because they're talking about it being newer and bigger and causing problems, which is the same narrative we get in the formal scientific literature a decade or two later. But why people started using that phrase, we don't really know. If decades later, naturalists start saying, well, of course, it's not really from Norway, but people called it the Norway rat because X, Y, and Z. But I kind of get the impression that they're speculating about where the name came from.
Mm-hmm.
And actually why why people started calling it the Norway rat, I don't know. But it seems quite plausible that it's something that in common English took on, and then that spread to other countries afterwards. Although that said, there's a Dutch natural historian in 1735 who quite possibly has the first formal scientific entry on brown rat in one of his texts, Albertus Seber, and he calls it Mus ex Norvegia.
Hmm.
And he gives no explanation as to that name at all. It's a very short entry. It has a picture, which is why we can be fairly confident that he's not referred to. So some people think there's been a confusion with the Norway lemming.
Oh, okay. Those look pretty different from rats.
They're very different, yeah. But the confusion comes in because Linnaeus actually missed the brown rat. Linnaeus doesn't describe the brown rat at all, even though other people writing around the same time in Europe had, which is possibly because it hadn't reached Sweden at that point, which is where he was writing. Although, of course, he does describe many species that were not present in Sweden as well. But he does refer to the Norway lemming, what we now call Lemmus lemmus. And because at that point everything was kind of lumped together under Mus, he calls it Mus lemmus. Sorry, Mus norvegicus, rather. So he does have a Mus norvegicus, but it's actually the lemming. And some people looking back on that later have said, maybe this is where the confusion comes from. Maybe that's where I don't think that's the case at all, because Albertus Seba has a picture of what is clearly a rat. You wouldn't want to be sure exactly which species, but it's not a lemming. Lemmings basically don't have a tail, and rats have a big long tail, and you can see this is not a lemming. But he gives no explanation about why it is Musex norvegia. So that would be the mouse from Norway.
Yeah.
Who knows? Maybe because that was already the phrase being used in English, maybe also in Dutch outside of scientific circles.
Yeah. I will say that this reading has really, well, I don't know if it's really helped me, but it has helped me identify the rats living around my around my neighborhood as brown rats. I guess I had never really thought about there being different types of rats. We've got a pretty healthy population living in our trash bins in my building, and I feel like I'm normally like pretty even keeled. But the other day I went to throw out some trash and I opened the bin and a rat saw me and it screamed. I screamed. My dog came running because she's a little bit of a ratter and she was so excited.
We had a rat burrowing up underneath our kitchen a couple of years ago. It's an extension, so it's kind of where the extension meets the main house. It had found a way in and was cutting a hole through the mortar. We saw its little nose poking out, and our dog just wasn't really interested. She came in and had a look at it, sniffed at it, and went away again. Which is just like, what do we keep you around for?
I mean, not to sound like I live in the dirtiest place of all time, because I really don't. But my dog's that way about. They're called American cockroaches, the wood cockroaches that are about like 4 or 5 inches long. I don't think you guys have them over there.
No, fortunately.
She'll see them and they're pretty slow and they don't want to be inside. They mostly come inside when it rains because they get confused. But she'll see them and she'll just like slowly follow them around the apartment. I'm like, please kill it. And she's like, no, I only kill pigeons and rats. But anyway, this actually also gets into something that you're studying, which is that because rats follow human populations, it's interesting because like I've kind of always thought about like human populations doing like these ebbs and flows, but like you have these two different populations of rats and they're also having kind of like their own miniature war across Europe for who has dominance. Can you kind of talk a little bit about that? This is also one of the main focuses of your project, was like the black and brown rat kind of existing in the same space and how that panned out.
Yeah, well, actually, so our interest in brown rats, I've got super fascinated in this recently, as you can probably tell from some of my answers. But initially, the reason we're interested in brown rats is in order to get a better understanding of what's going on with black rat ecology in previous periods. So when it comes to the black rat, we know that it flourished in much of the Roman Empire, for example. When the Roman Empire evolved, broke down, ended, collapsed, you choose your preferred phrase, it's quite contentious. But when it's certainly in places like Britain, up in the northern provinces, the system fell apart quite a lot in material terms. Urbanism declined significantly, if not completely. Trade declined massively, particularly bulk trade, so trade in perishable goods and the kinds of things that rats will eat. And rats disappear at that point. So from the perspective of somewhere here like York, we're up on from a Roman point of view, we're up on kind of in the cold northern wastes. And we have rats appearing even really quite early on. Even north of here, up on Hadrian's Wall, they have rats turning up in the grain stores at some of the forts and that kind of thing. When the Roman system collapses or ceases to apply, in a British context at least, the rats seem to disappear pretty much straight away as far as we can tell. They then come back again centuries later, and they're absolutely thriving by the late medieval period. But there's good reason to think they're first of all popping up here and there back again during the Viking Age, which, yeah, I mean, it's kind of a dream when you're trying to sell the story to kids or something, especially in a town like York, where we have a lot of tourism connected to Vikings and to some extent to Romans as well, and we can tell the story of, the rats came with the Romans and then they went, and then they came again with the Vikings. But we're really interested in what are the processes actually underlying that. So it's partly to do with things like urbanism and trade networks and so on, but it's got to be more complicated than that. And one of the things to remember with rodent species is they live fast, they die young, and they leave thousands of corpses. But most of those never survive for us to look at. So they'll, they breed extremely fast. They can breed several times a year with litters of maybe like 7 or 8 each time. So they could reproduce exceedingly fast, but most of them won't survive to a year-old.
Yeah. I was reading that their average gestation period is 21 days. Mm-hmm. And then their average lifespan is 3 years in the wild, which to me seemed even kind of high.
That is longer than most estimates, but it will depend on the species and it will depend on the context where you are in the world and so on.
Yeah.
But it's
But I mean, 21-day gestation period and 5 weeks to sexual maturity, like it's a lot of pups.
yeah, and if the resources are there,
Mm-hmm.
if there's the space and the food for them, the population can explode. If the resources aren't there, it crashes back down again, and most of those pups will starve, basically. So the populations fluctuate like mad. Now, if you're looking at a big modern city it's fairly stable, there's an abundant supply of food, you're going to have a lot of births and a lot of deaths, and the population's going to stay fairly stable. If a city block gets leveled for a new development, there'll be a local population crash, and then when the resources come back, it'll feed back in again. But if you're looking at an early medieval town, which is a much, much smaller operation, which is probably also, in some respects, less urban, has less of the characteristics we associate with rats, they're also going to be competing with local species, so the species that live in the surrounding area, and that might not do so well in an urban setting, but they're just there in the hinterland, and they can keep coming back in and repopulating. Whereas rats are reliant, if they die out, they're reliant on another ship bringing them in again. So you've probably got this really, really dynamic situation with populations kind of booming and busting and going completely extinct in some cases, and then getting repopulated or being topped up by new rats coming in on ships. It's all going to be extremely dynamic. So when we start to see them kind of popping up back again in Viking Age settlements, we have this question of, are they really established? Or are they coming in on ships, living for a while, dying out again? By the late medieval period, they seem to be ubiquitous. So maybe not in the far north, maybe not in in Norway. You only find them in the big port towns, but in much of Europe, they start turning up all over the place. Not only in towns, much more common in towns, but even in smaller settlements. Because in fact, we see this in the Roman period even. We find rats in Roman villas. We find them in Roman forts. They're not urban per se, but they're well-connected sites. And the question is, there are rats from a Roman villa near Oxford, a place called Gatehampton. It's just up the Thames from London. Plenty of rats there, but would they have been able to survive if there wasn't communication up the Thames? If there weren't supplies being brought in and so on. So it's all about networks, we think. And we're trying to understand this system. We're trying to kind of calibrate it. And we obviously work from what we know about black rats in modern ecology, but they're more or less extinct in Northern Europe now. So you have to look and kind of compare to what's known in other places, or to historical records where they're competing with brown rats. And in any case, the kinds of settlements that existed in the Viking Age don't exist anymore. There is no direct comparison. So it's always this kind of bootstrapping process of, we know a bit about how this species works in some places. We can't port that straight to medieval Europe. We have to actually try and kind of look at the data, see where they turn up and where they don't, and so on. But one of the huge challenges is, because all of our observations of black rats in Europe come from, or formal scientific observations, come from a period where they're being outcompeted by brown rats, at least in the north of the continent. So one of the original reasons why we decided to look into this whole kind of replacement thing was to get a better idea of how much has changed. And one of the reasons in turn for that, completely going off on a long tangent, but one of the reasons for that is that there's a huge amount of argument surrounding plague, surrounding the Black Death, that is about, oh no, you wouldn't have found rats in that kind of settlement. We know black rats. We know they only thrive in towns, so they can't possibly have killed hundreds of thousands of people in rural contexts.
Yeah.
But of course, we don't really know that.
So the idea being that, I think the common idea being that black rats were a reservoir for Y. pestis, which is the creature that causes the plague, the black plague, the bubonic plague. I was looking through Google Scholar articles, and there's also debate whether the rats were even the primary reservoir, if it was actually just humans instead.
Yeah, so it's complicated.
Yeah.
It's complicated and it's really contentious. So yeah, the traditional model here is that, and reservoir is maybe not quite the right word. The first thing to say about plague is it is a rodent disease primarily. It's a wildlife disease, at least.
Mm-hmm.
Probably from Central Asia originally. It lives wild there in a variety of rodent species that have quite substantial immunity to it, so they're not dying of it on a large scale, but it's just kind of circulating. And it will occasionally spill over into human populations, quite possibly via species like rats, or via people actually going and you're hunting marmots or that kind of thing and bringing their skins back and getting infected that way. When it spills over into human populations, the big question becomes, how does it then spread away from those original source regions? And the traditional model is that that's all about rats. And that's partly because rats were observed to be able to spread plague via their fleas in at least some places during the third plague pandemic, which is the late 19th and 20th century outbreaks of plague. Incidentally, the era in which plague spread to North America. So plague is now endemic in parts of the southwest there, I believe.
Yeah.
It wasn't before that particular pandemic.
Yeah, if you go camping in California, there's signs saying not to touch the chipmunks because they carry like leprosy and plague, which is good. I mean, you shouldn't touch chipmunks in general, but yeah.
So that's a fairly recent development. But yeah, chipmunks are closely related to some of the species in Central Asia that plague has been circulating in for probably thousands of years. So the question then is, when the Black Death in the mid-14th century sweeps across Europe, killing huge numbers of people, what is allowing it to do that? And then traditionally we thought, well, there are rats moving with the people, there are rats in all the settlements that the plague can get to, allowing it to kind of thrive there and so on. And the model goes something like this. So rats become infected via their fleas, via rat fleas. There's a specific species of flea that thrives on rats. The fleas, this is quite gruesome actually. It's quite grotesque. The effect of the plague bacterium on the fleas is to block up to cause them to create a film that blocks up their digestive tract, so they can't really swallow, they can't ingest. So then you have fleas which are starving because they can't actually ingest blood. And what do they do if they're starving? They just keep on biting. So they'll bite and bite and bite like mad. And in the process, they'll keep spreading the blood that they're unable to ingest into, across all the different animals they bite. And that's fine as long as it's just rats. Well, it's fine for us as long as it's just rats. But if the rat population dies out, maybe not the whole population, but if the rats die off substantially because of the plague, then those fleas will potentially jump to humans. They'd much prefer to be eating rats, but they'll jump to humans if they need to. And at that point, it spreads into humans. So that's the traditional model. And I don't think anybody denies that this does happen, that it can happen in the right setting. The argument is about whether this is actually plausible as the kind of the primary way by which it spread across Europe in the 14th century and then in successive waves right through the 15th, 16th, 17th, even into the 18th. Alternative ideas involve, spreading via... Well, plague can spread directly from human to human, but that gives us what's known as pneumonic plague, which kills very, very fast. So plague can go airborne, effectively. If plague gets into your lungs, you can cough it out. And if other people catch plague that way, it's basically moves straight to the final phase, and you'll die very fast, which makes it very difficult to conceive of that managing to maintain long-distance spread. Maybe in today's world, that might be a problem with people moving much greater distances much faster. But if you're going to die within a couple of days, you're not likely to take it to the next town.
Mm-hmm.
So the suggestion then is that maybe it's able to spread in ectoparasites on humans themselves, so lice primarily. I should say I'm not really qualified to evaluate that hypothesis, but that's kind of where the argument goes at the moment. Is it all about the rodents and their fleas, or is it about human-to-human transmission via human parasites instead? Which some have argued is more consistent with the speed of the spread and kind of the dynamics of it and so on. We're trying to be open-minded on this. We're trying to enter it. It gets a very contentious debate. There's some scholars who can get quite heated about this, and we're trying not to incur anybody's wrath until we actually think we have any kind of an answer.
It's definitely interesting reading the papers because it's just like a very polite slap fight. I don't really know how else to describe it.
There are one or two papers where it gets less polite, in fact, but mostly it's polite. That's true.
But you can tell people feel strongly about it. It's definitely it's an interesting jaunt in literature if you're curious. It's an interesting topic, so I think they're definitely worth reading.
I was going to say, one of the big developments lately in this is that people have started being able to extract plague DNA from human remains from plague bits. It's something you've quite possibly covered at some point on your podcast.
No, that's really cool.
Well, you should do. You really should do. It's a an amazing topic. So it's been maybe about 20 years since the first paper doing this, but in the last 5 or 10 years, it's really taken off. And not only have they managed to recover plague from the teeth of human plague victims, and therefore prove that the disease we know from the history books is actually the same bacterium that we know today, which was not, even that was contentious 20 years ago. So not only have they proven that it's that disease, they're also able to plot the genetic tree, as it were, of plague itself and see how the different strains have evolved through time. And one thing we now know from that is that, so the traditional idea was, plague spreads in the mid-14th century during the Black Death. It causes havoc, it dies out, and then, 10 years later, it comes back again and causes havoc again. It's always spreading in from the east, which is something that there's a historian, Nuket Varlik, she's a historian of the Ottoman Empire, and she writes a lot about plague and disease in general. And she calls this epidemiological Orientalism, because it's this idea that, all these bad things keep coming from the east. It's quite problematic when you think about it.
Mm-hmm.
What we now know from looking at the phylogeny, looking at the genetic tree of plague, is that every plague genome that they've recovered from a human in Europe from the entire second plague pandemic, so that's the Black Death right through to the early 18th century, they are all descended from the Black Death. They're not the same strain. There's all kinds of different substrains and things going on, which I'm not on top of enough to go into in any detail, but they're all descended from, which suggests that that there is some kind of a reservoir, so a long-term reservoir of plague somewhere, broadly speaking, within Europe. Now, it doesn't have to be Europe per se. It could be in the Caucasus, Western Asia, and so on, but somewhere away from the original source region. And that changes an awful lot of stuff. So part of the question now becomes, okay, not only how was plague managing to hang out in Europe for centuries while only occasionally causing these major outbreaks, but also where did it go? Why did it die out?
Yeah. And then how did it come back? This is super interesting. And I do want to get to just like a few other things about your study before I lose you, because we're running short on time because you're so interesting and you have so much to say. One of the really cool things about your study is that you're combining a lot of different techniques. So zooarchaeology, so looking at the bones, which we discussed. You're also doing ancient DNA sequencing on rats, which is really cool. And kind of related to that, you're also doing like molecular species ID. And you're also doing radiocarbon dating, which you'd mentioned earlier, to like kind of figure out when they're from. And then on top of that, you're also doing an isotopic analysis, which is something that we have covered like pretty extensively on the show to understand diet in rats. And you're also doing documentary evidence. This is just like a huge range of methods. And I think it's incredible. And also like the idea of doing ancient DNA and molecular species ID on rats, like had never really occurred to me, but it's super cool.
Yeah, I mean, it is a challenge given their small size. And you don't want to go destroying all the evidence. And these are destructive techniques. So we have to be quite systematic and quite considered in which bones we do sample and what we sample them for. And obviously, we have our own ethical concerns there, but we also have curators who we need to persuade quite rightly that the destructive analysis we're going to do is justified. That's quite easy when you're working on a cow, where the bones potentially weigh 500 grams or something.
Mm-hmm.
Typical rat bone, well, you're lucky if it's half a gram.
Right.
But we could potentially run all of these techniques on one specimen, but there won't be a lot left of that specimen. So you have to be a bit careful. And again, 10 years ago, I don't think it would have been possible. One of the absolute game changers here is the proteomic identification. So it's a technique that we call zooms.
Mm-hmm.
For some people love that name, some people hate it.
It does make it hard to search because of the company.
The technique was the technique had taken off before the company did, but
yeah, no, that's totally fair. If you're looking for a way to search it that's not zooms, it's collagen peptide fingerprinting. We'll bring it up for you.
So what this is a very cheap, basic, rough and ready way of identifying proteins to a different species. We primarily use it in our project. We exclusively use it on bone collagen, which is what it was originally developed on. People have also made variants of it to work on different proteins that survive in eggshell, for example. But primarily we use it on bone collagen. And this is the main constituent of bone. So if you've talked about isotopes in the past, you've probably mentioned that mostly what you're measuring the isotopes in is the collagen within the bone, so the protein component within the bone. It's something like 40 to 50% of the mass of a typical bone. And it survives really well. It survives much better than ancient DNA. There's much more of it to begin with, and it is more resistant to damage in the ground. What we do with Zooms is stick that in an enzyme, so extract it in a broadly similar way to what we'd do for stable isotopes, get rid of the mineral component of the bone and just get out the protein. We put it in an enzyme, trypsin usually. Some species, they use a different enzyme. And that cuts it up in predictable places. So in theory, we could take that protein and we could sequence it. We could run it on a machine we have called the Orbitrap and actually get a full protein sequence. That would cost us a lot of money. And it would use a bit more bone as well. This method, we take the smallest little piece you can imagine. So 10 milligrams of bone is usually plenty. And we just run it through the enzyme, cut it up in predictable places, and then run those through a very, very fast form of mass spectroscopy called, we use a multitof. There are other types people use as well. And that shows us the size of those predictable chunks. Now, each one of those chunks is a string of amino acids. And if we're lucky, the species we're interested in have different sized chunks. So they have differences in their sequence, which means the trypsin will cut them in different places. In our case, we're lucky because black rat and brown rat have one marker difference between them. So likewise, sheep and goats have one marker difference between them. And that's been a huge game changer in zooarchaeology because telling sheep apart from goats is, it's a cliche, really, separating the sheep from the goats and all that. But it really is a challenge, and this technique has made it much more feasible. For us, it's the same kind of thing. It would be really difficult to do this project if we couldn't be sure which of our specimens were black rats and brown rats.
Yeah.
If we have a skull, we can tell them apart. Anything else, we can have a go at telling them apart, but we can't really be confident. Now with Zooms, we can take a tiny little shaving off it, and we can get that answer. Now, of course, ancient DNA will also give us that answer, but we'd have to cut off a much bigger chunk of bone, spend a lot more money and a lot more time to get that answer, and it might turn out that actually we're not interested in this bone because it's the wrong species. So Zooms is, I say, an absolute game changer. It's really the thing that makes this project possible. And it's a technique that was, I should have to plug York, my university. It's a technique that was developed here about 2009, 2010. The first papers came out on it. And now there are lots of labs across Europe and North America and some in China as well doing it. So it's really kind of become established. But we're really, really lucky here in that we have a unit within the department. It's called BioArch. So for biology, archaeology, chemistry is the concept behind the name. So yeah, so we're very lucky here in York in that we have this unit BioArch that I'm part of, where we have expertise in most of perhaps all of the major techniques that people use within. There's a slight difference in terminology. So bioarchaeology in Europe refers to biological forms of archaeology. Bioarchaeology in North America primarily refers to human bone analysis. So kind of distinction. But we basically do pretty much all of the major techniques within bioarchaeology here. We have an ancient DNA lab. We have our Zooms proteomic facilities, and in fact more advanced forms of proteomics as well. We do stable isotopes. We also do things like lipids and other residues of pottery, which is not so relevant to my project. And of course, we have the zooarchaeology labs and so on as well. And we have human osteologists. It's kind of, we can do everything in-house, which is everything apart from the carbon dating in-house, which is really, really nice. So yeah, the first thing is usually the Zooms. It allows us to get a species ID quickly, cheaply, and definitively. It's perfect. The ancient DNA analysis tells us a lot more. So it's really just, you could consider it the core of the project. I mean, really, I guess the basic zooarchaeology is the core, but without the ancient DNA, there would be much less of a story. And what we're trying to do here is, well, primarily tracing routes of dispersal, picking up where a species is, where rat populations have died out and been replaced. So I mentioned earlier on, in England, for example, in fact, in much of Europe, we have rats in the Roman period, then they disappear, they come back again some centuries later. Some earlier ADNA work we did kind of before the project got started properly, this was a collaboration with various other groups, showed that when they come back, it's a new population. So there's always been this question about, did they really die out? Or was it just that we don't have much data from the intervening period and, the population recovered later and became more abundant again? Well, that would be quite plausible. But as it turns out, when rats become common again later on, the medieval period, they're genetically different.
That's really cool.
Now, as things stand, yeah, it's really nice. So this was a paper we published in 2022, led by a researcher in Germany at the time. She's in China now, called He Yu. I know that was really nice starting point, but we didn't have that much. We didn't have that much data within it. So we saw this big turnover, but we didn't have enough kind of reference data. We didn't have enough samples from the Mediterranean region, for example, because it was the first time anyone had ever looked at black rat aDNA, black rat ancient DNA. So what we're going to be trying to do on this project is really flesh that out. So when we see they're coming back and they're coming back from somewhere different, can we pinpoint where they're coming from? Can we look at the routes they spread along the way? Can we look at, Viking Age rats in England versus Viking Age rats in Sweden and see, comparing them to specimens from Central Europe and to Ukraine for that matter as well, and trying to get this kind of, see what are the routes by which they could have spread. And we may also find surprises. So in this case, we already had good reason to think that there was several centuries gap and then a new wave of colonization. But there may be times when that's happened and we're not aware of it, because the kind of resolution we usually have on our dates archaeologically, if a species died out somewhere for 50 years and they came back, we'd have no idea. If that happens and it's repopulated from a different source, genetically we might pick that up. So there could be all kinds of surprises here. And rats are perfect for this because, if you think about the things people have done looking at human migrations and expansions via ancient DNA. Now, humans are great. Don't get me wrong. There's nothing wrong with humans. But if you want a species that's going to give you high resolution on movements and connections, humans are rubbish, right? We live too long. We have too few children. Too many of them survive. Okay, so there's not as much kind of resolution there. Rats, on the other hand, as we were talking about earlier, they have very short generation times, typically 2 per year maybe, and they have very high selection. Most of those offspring will die. As a result, they can evolve extremely fast. They can accumulate differences, which we can then use to actually pick up, to give us resolution spatially and to give us resolution temporally in the patterns we see. So in theory, at least, they have enormous potential. And we'll see how much we manage to make that materialize. So that's the core of what we're doing with aDNA. There is a kind of stretch goal, which we'll see if this works or not, and that's to see if we can actually pick up evolution in the rats themselves. So not just using them as, looking at differences that show us about different populations branching and splitting and that kind of thing, but also looking at directed evolution. And in particular, whether we can see a reaction to plague.
This is so cool. This is really cool.
I think so. It's nice when other people do. But yeah, so this traditional plague model relies on the rats dying. The whole idea of the fleas jumping ship relies on not only on rats getting infected, but rats dying of plague. Now, if rats are dying of plague on a large scale, and given how rapidly they can evolve, short generations, high selection, and so on, we should expect to see some selection in their immune genome. So even if they don't become completely immune, we would expect to see that, not everybody dies of plague, not every human that gets plague dies of it, not every rat that gets plague dies of it. There will be genetic characteristics that favor survival. It's going to be fuzzy, it's not likely to be an absolute thing, and there will be genetic characteristics that don't. So we should expect to see selection in the part of the rat genome that is related to immunity. And there are modern rat populations in some places, Highland Madagascar is the famous one, which are quite substantially resistant to plague. They've not been all that well studied genetically, so it's not like we can point to a gene and say that's the plague resistance gene or anything like that. But if we can see that at the point when we know there are big plague outbreaks, there is evidence of selection in that part of the genome, then we have quite good circumstantial evidence that they are being affected by plague in large numbers. And at that point, if we can say that, okay, we know the rats are in these towns, we know they're dying of plague, we don't actually know for sure they had the right fleas. That's the big outstanding question, and that's not one we're going to be able to answer, but somebody else might. But if we assume they had the right fleas present, at least, then it's very hard to imagine that was not having an impact on the spread of plague. Now, it doesn't mean it's necessarily the whole picture. And again, a lot of those headlines you're talking about, these kind of contentious articles arguing with each other, it often feels a bit like it's people shouting, it's all about plague, it's all about rats, so it's nothing about rats, so on. In reality, it's likely to be somewhere in the middle, and that we know that this species can, again, caveat, with the right fleas, spread plague, and we know it was present in many of the places that got plague. It seems unlikely it didn't have some impact, But the question is, is it an aggravating factor or is it a critical factor? And we really don't know. We might provide some good circumstantial evidence for it having been quite systematically involved.
That's so interesting.
But it's a bit of a stretch, though, because it does depend on us actually being able to get enough resolution on the right bits of the genome. So, yeah, we'll see.
Can I ask you just like a practical question, which is when people are digging up archaeological sites, where are they finding these rats? And how do they know that the rats are, because rats burrow. So how do they know that the rats are from there and then? Does that make sense? Are those good questions?
They are absolutely the right questions. Like, this can go kind of two ways.
Good.
So there is a danger of just assuming, of trusting things too much, saying, well, I've got rats from this 9th century settlement, so they must be 9th century. That's all good. If anything, there's more of a danger, or there has been more of a problem, that a lot of zooarchaeologists, a lot of archaeologists over the years have said, rats, they burrow, these must be intrusive, we'll throw them away, or at least disregard them, maybe not record them. So there is this kind of problem. And yeah, it is a huge problem, particularly for brown rats, but both species burrow. So we always worry about small bones anyway. So the way like the formation of archaeological sites works, there's usually some disturbance, there's some mixing. Small bones in particular can kind of filter down through cracks in the sediment. They can travel effectively. That's always a problem.
Mm-hmm.
When it's a species that actually actively travels as well, that actually digs holes, then it's always a huge concern. So there's a few different approaches to this. One of them is just to say, okay, the signal is going to shine through the noise. And we're doing that to some extent. We're mapping out all the data we can find from the literature as well as from our own research. We're seeing the patterns. Where things stand out from the patterns, that's when we have to make a call. So at that point, it's, okay, is this an outlier because it's intrusive, is the word we use, because it's dug down into the level from a later period? Is this an outlier because there was something unusual going on here? And at that point, the next thing you do is you read the report very carefully and you see what kind of a context is it from? Is it from something? If it's from what's a good example? Actually, the first ever wasn't technically the first ever. One of the first ever publications about pre-medieval rats in Britain is a good example of this.
Mm-hmm.
So, and up until the late 70s, we thought that rats had come to Britain in the medieval period. So the cliche was returning crusaders coming from the Eastern Mediterranean brought rats with them. Again, everything comes from the East, right?
Of course.
That was the traditional kind of narrative. And then people started to find them in Roman sites because they started to sieve more on those sites. And people actually started to pay attention to the small bones, and suddenly it's, oh, okay, we have rats in the Roman period. Something more complicated is going on. One of those sites was here in York, a site called Skeldergate, published in 1979, and it's a well. And these rats are really down at the bottom of a well, in a layer that's full of rubble that seems to be from the late 4th century. And in that kind of context, you can be reasonably sure they might not be from exactly the same period as the bits of pottery you find in that same rubble. Maybe they dug in a little bit later. But you can be pretty sure they didn't dig down like 3 or 4 meters from the top.
Right.
Yeah, exactly.
The top. Yeah.
So looking at the context is an important thing. And when it comes to looking at the other thing, of course, is the dating. So if when all else fails, when we can't be confident, when it's important, when it looks like it's a sample that might change the story or a sample that we're going to rely on in the genetic study, and we can't be 100% sure about the context, then we get a direct radiocarbon date on it. And we have the budget to do that on something like 150 specimens. So we can't do it on everything, but we're able to go and check things where we need to. Of course, that has some of its own problems, which are perhaps too technical to get into now, but particularly when we get into later periods. So effectively, you can't really carbon date anything after about 1650, or at least between about 1650 and 1950, you just get a very vague answer.
Mm-hmm.
So when we're looking at the spread of brown rats, we have a much bigger problem. For one thing, brown rats burrow much more than black rats. It's really what they're known for. The cliche is that brown rats are tunnelers. You find them in the sewers, you find them in the basement, you find them in the garden. And black rats are climbers. In a wild setting, they're arboreal. They kind of evolved living in trees, and now they live in attics. That's the cliche. It's a little bit more complicated than that. But it certainly holds true generally archaeologically. When we find a rat that has burrowed down into an earlier level, and we check it and we date it and realize that it's actually recent, it's almost always a brown rat.
Yeah.
Black rats, maybe the dating's not quite right, but they tend not to be radically wrong. Looking at a site from the kind of 8th, 9th, 10th century in Estonia, brown rats, you date it, and it's from about 1970 or something like that. We had one from Portugal, one from York, various things where, okay, it's a 10th century context, we've got rats in it, we do zooms first, the zooms comes back to saying it's a brown rat, and immediately we think, okay, right, that's probably burrowed in. Now, that's not a huge problem when we're looking at black rats, because we can do the zooms. We can check the species, and if it's the wrong species, we're immediately suspicious of it.
Mm-hmm.
But it is a problem when we actually want to study the brown rats.
Right.
And it's a particular problem because carbon dating won't really help us. So carbon dating will tell us if it's before or after the nuclear tests in the 1950s, 40s and 50s, because that massively changed. I don't know if you've done radiocarbon dating on your podcast before.
Not in a while. It's probably worth it. Yes, it's probably worth explaining why you suddenly brought up nuclear tests.
Yeah, so carbon dating relies on the knowing the amount of radiogenic carbon-14. So carbon-14 is an isotope of carbon which occurs naturally, but it occurs naturally based on radiation, cosmic radiation, technically. Nuclear explosions massively increase the amount of carbon-14 in the atmosphere. It's fluctuated anyway naturally through time. And of course, it decays over time. And when we're dating things normally, it's a matter of looking at the rate of decay and then adjusting that based on the known fluctuations in the starting point. So
mm-hmm.
In the early days of carbon dating, we just looked at the decay. We assumed there was a constant amount in the atmosphere, and that gave a lot of wrong answers. And then people realized you needed to calibrate this. And I could talk for half an hour on how they went about doing that. It's a really interesting topic, but I won't do that now. But we have a pretty good understanding of it. The trouble is that for the last few centuries, perhaps this is because of industrial revolution of fossil fuels. Maybe I'm not sure on that. But for the last few centuries, it's fluctuated like mad, and it makes it really difficult to get a good date. Then when the nuclear tests happen, you get this enormous spike, and it's been gradually settling back down to normal for the past few decades. So anything that is if you've got a rat that's from the 1960s, you might be able to get it down to a 2-year period. If you've got a rat that's from the 1860s, you're probably going to get it to a 3-century period.
Yeah.
and before that, results vary depending on kind of exactly where you are in time. But it's a huge problem for us because we're trying to pin down how this species spread across Europe, and indeed to America as well, actually, around the 18th century, right in the middle of this period where we can't get a good answer from radiocarbon dating. So what can we do? We can't trust the context most of the time because they burrow. We can't trust the radiocarbon dates because it's all over the place. And we have two answers to this, really. One of them is to look at rats from shipwrecks. Because if we have a shipwreck site, well, we often have an exact date. They're often historically documented. We know when it went down. Even if they're not, we probably know roughly when, based on the material culture on board, based on the artifacts on board. And we can be pretty confident that a rat won't have swum to the bottom of the sea, burrowed into the wreck, and then died there. So any rats we find on board, they went down with the ship. And this is an approach that has really been pioneered by my colleague Eric Geary, who's the guy who led the Ratting of the Americas paper, which included some of these early shipwreck sites. So that's one way we can do things. Trouble with that, of course, though, is it tells us which rats are aboard the ships, but it doesn't tell us so much about which rats are established in populations on shore.
Mm-hmm.
If we know the history of the ship, if we know its itinerary, where it was launched, where it went to, maybe we can piece together a story. But it's not ideal. So the other big thing we're doing is going to natural history museums and looking for museum specimens of rats. So rats that have never been in the ground from as early as we can get. And so far, some of the earliest samples we found are back to about the 1830s. It would be great to get things from the 18th century, but that's probably going to be impossible. I mean, we know that European naturalists were collecting things like mad in the 18th century. We haven't yet managed to locate where any of those big early collections ended up. Whether anybody would let us sample them is also a big question. But we have managed to get permission to sample quite a lot of 19th century samples, 19th century rat specimens. And that kind of bypasses all the problems of archaeological dating. So yeah, I'm really excited about this. In fact, this wasn't written into our original project plan, but it was an idea that we came up with It's quite an obvious idea when you think about it that we probably should have written in. But yeah, that kind of gets us around the dating problem.
Yeah, no, it's really interesting. I was also just curious, do you think people are better about keeping rat remains now? Because I feel like I can envision a past where people found rat remains and were like, this is useless and just tossed them.
Yeah, I think people are much better now. And I think even in the past, most often things probably weren't thrown. Well, I mean, yeah, there are always horror stories about sites where they threw all the bones away, that kind of thing.
Yeah.
But I think more often it's a matter of people not actually studying the things.
So everything being collected, but then just not looked at.
Yeah, or perhaps again, identifying rodents is considered something of a specialism. Now, actually, rats are pretty easy. They're bigger than most rodents, and in Western Europe, at least, there's only 2, 3 species in that size range, so they're actually really quite easy to identify. But people don't necessarily realize that if that's not what they're used to working on. So a lot of the time, they might just get lumped together as rodents or small mammal, and so they do get published. They turn up in the report,
Mm-hmm.
but they turn up in the report in a broader category. Another thing is how much people sieve. This is a huge problem. So in some areas where archaeologists don't normally do much sieving, we've found rat remains by talking instead to the archaeobotanists.
Ah,
Again, I don't know if you've ever
that makes sense.
have you ever covered archaeobotany on this?
I don't think we've had someone who does straight archaeobotany. We've had quite a few people who do, we've had a lot of teeth people who look at the little, oh, my brain's not working, but the little plant remains that are stuck in teeth. But I don't think we've talked to an archaeobotanist that's their main job.
Okay.
But I guess I need to bring one on.
Yeah, good. I mean, I could recommend somebody, but
I would love that.
we can sort that out afterwards, I guess. But so the archaeobotanists obviously study plant remains. Most plant remains that you might find on an archaeological site are exceedingly small, so things like individual charred cereal grains, that kind of thing. Therefore, they basically don't have any work to do at all unless there is an intensive program of sieving, usually what we call wet sieving or flotation. So you take a bucket or two of soil and you run it through a machine that sorts the stuff that floats from the stuff that doesn't float and catches both of those on a fine mesh. And then you kind of painstakingly sort through it with a pair of tweezers and pull out all the plant remains and put all the bone remains on one side. And a lot of the time, depending on where you are, what country you're working in, what tradition you're working in, a lot of the time those bone remains will just sit on a shelf somewhere, never get looked at. In a lot of places they will. So here in Britain, we will, on a commercial like a CRM site, this stuff will, if it's important, if the context is considered worth looking at, then the bones and the plants will systematically get looked at. These days, wasn't necessarily always the case. But there are other places where, the zooarchaeologists mostly look at the bigger stuff that's come from what we call the bulk, and the bones from the heavy residues, as we call them, so the bones that sink out of the archaeobotanist samples, don't necessarily ever get looked at, or it might be a long time before somebody thinks to look at them. So, for example, we had a bit of a hunch once. There's a late antique, so kind of late Roman and into post-Roman settlement in Albania called Butrint, which has been subject to a lot of excavation. There's been a lot of kind of big international projects there over the years. Lots of zooarchaeology reports, but none of those zooarchaeology reports really have any micro-mammals, as we call them, so rat-sized or smaller things. And I noticed that the archaeobotany report for one of these projects had been done by a colleague of mine who's now in Barcelona, who I worked with briefly many years ago. So just like on a hunch, let's write to her and say, do you have any small samples? Did you have any small bones in your samples? It turns out she did. They were still here in England, in fact, where she'd previously worked at the University of Nottingham, and one of our team went down there, had a look through them, and yeah, they have rat bones, which is exactly what we were hoping for.
That's so gratifying.
Yeah, we're in the process of getting a carbon date on one of them at the moment, so we'll see.
That's very exciting. Yeah, that's. I was just thinking a lot about like how sites are managed and like how you pull stuff up and like finding these very small bones, especially like. I think that I know that this is a bias of mine. Like if we find humans and like we're very careful and we look for every remaining piece and we'll find like little teeth and whatever, but people aren't really looking for rats in the same way. Like if they find evidence of rats, they're like, okay, rats. It's an interesting like shift in perspective for me because I'm very human centered.
I mean, yeah, there's an image problem for one thing, but it's not obvious It's not obvious why they would be relevant.
Yeah.
And partly because, 90% of the time when zooarchaeologists are looking at micromammals, they're probably working in early prehistory, and they're probably using them to look at reconstructing the environment around a site. So, if you're working on a Paleolithic cave site, yeah, you keep all the micromammals, and somebody will study those, and they'll use it to talk about, well, it looks like in this phase we've got evidence of some kind of avian predator hunting in this kind of an environment outside the cave, and the fact that they're doing that means probably it's not occupied at this point, and so you get all these kinds of analysis that are very paleo-environmental.
Mm-hmm.
And that's all important stuff in that context. Then if you move to a medieval city, people find a few bits and pieces. If you're thinking small mammal remains, well, they're useful for reconstructing the environment, then you think, well, yeah, that doesn't really apply here in the middle of, I don't know, medieval Paris or something. And so it's not obvious why they would be of interest. But hopefully we're showing that some species at least are of interest.
Yeah, no, totally. I mean, you sold it to me. You definitely sold it to me. I feel like the idea of understanding, not to bring it back to the people again, but the idea of understanding humans via rats is brilliant. And I'm really excited to see the stuff that comes out of your study. Speaking of which, I think that we should probably wrap up so you can do other things with your day, like see your children. Can you tell me, if someone wants to learn more about either your project or about zooarchaeology in general, where's a good place for them to start?
Okay, well, zooarchaeology in general. I wasn't thinking about zooarchaeology in general when you briefed me on this before. Let's start with my project then.
Okay.
We do obviously have a website. It's a very basic website, but if you search for Rattus and York, you should find it. That has some of the papers we've published recently, and it has some basic blurb and so on. So a couple of papers we've had out, neither of which they're both collaborations with other people, but they should be pretty accessible, and they give a pretty good introduction to some of the things we're looking at. So the first one is the one you mentioned earlier, the ratting of the Americas. Again, very much not my paper, it's Eric Geary's paper. We were just involved a little bit in our project. But Eric is doing in the Americas something quite similar to one aspect of what we're doing here in Europe. So it gives a really nice kind of idea of the kind of thing we will be doing in the coming years. The other one is a paper by Jason Munshee South et al., and I'm one of the et al., which is a review in Science, which is all about the evolutionary and ecological history of the brown rat. So it's quite nice because I'm as an archaeologist myself, I'm a little bit outside my comfort zone working from historical sources, but it's quite fun to be publishing historical sources in a science journal, kind of alongside colleagues who are ecologists, who are bringing in lots of ecologists and evolutionary biologists, bringing in lots of other aspects of understanding of this history. So those two places would be a good place to start, I think, and they're both the kind of thing that don't require a huge amount of subject kind of discipline expertise to understand, I think. Zooarchaeology in general, I can tell you which textbooks I recommend to undergraduates, but
that's perfect.
I always think a good place to start is Terry O'Connor's book. It's from 25 years ago now, 2000. It's just called The Archaeology of Animal Bones. It's a good cover-to-cover read. It's not a textbook in the sense of kind of a reference that sits on the shelf. It's a nice kind of introductory read. And Terry was my predecessor here at York. He taught me when I was doing my master's, and he did quite a lot of work on rats in York back in the day. Well, I'm not sure if they feature in his book or not. But
I think I actually have a copy of that sitting on my bookshelf.
Talking of which, actually, to tie those two things together, another book by Terry, he wrote a book called Animals as Neighbours, which is specifically about animals that live around human sites. So what are often called commensal or synanthropic animals, including rats, but also pigeons, cats, dogs, foxes, raccoons, all the rest of it. And that is, again, it's probably researched, it's got references and so on, but it's also clearly aimed at a broader audience, not necessarily within archaeology.
I will say the other thing about zooarchaeology is that y'all produce beautiful bone manuals. And these are going to vary depending on where you live, as you mentioned earlier. But if you Google zooarchaeology bone manual North America, you should get, or UK or whatever, wherever you are, you should get some like pretty good recommendations with beautiful photographs of the bones of the animals that live in your area. I think those are worth having because sometimes like you find stuff when you're out on a walk and you want to know what it is.
Yeah, definitely. And actually, when it comes to small mammals, there's a bit of a shortage of good ID manuals. What we often end up using are the same guides that people looking at that naturalists use if they're looking at owl pellets, for example.
Mm-hmm.
So if you want to identify small mammal bones, a good place to start is finding a guide to owl pellets or raptor pellets in the region you're in.
That's a really good recommendation. I wouldn't have thought about that. The second question for you is, what are you reading for work right now?
Okay, for work, well, so at the moment I'm desperately trying to wrap up a paper that is all about this. Well, it's on the one hand all about what we can actually understand from how rodents are popping up in different settlements and so on, and on the other hand, it's about trying to review the data from Northern Europe in the early medieval period and put all that together. And this is meaning I'm having to, on the one hand, dig into some kind of classic ecological texts, and on the other hand, some up-to-date thinking on early medieval urbanism and trade. It's kind of pulling me in two directions at once. So I guess I will say, on the one hand, MacArthur and Wilson's 1967 The Theory of Island Biogeography, which is an absolutely foundational text in much of ecology. And on the other hand, I've got Sven Kalmaring's book from 2024, I think, actually, Towns and Commerce in Viking Age Scandinavia.
Amazing. It's really both ends of the spectrum.
Yeah, it can be quite a headache kind of switching mental gears between these things from time to time.
Mm-hmm. Yeah. And then third question, what are you consuming for fun right now?
Oh, for fun. So reading, I'm reading a book called A Fire Upon the Deep, which is a kind of classic sci-fi novel, which is, yeah, it's good. I haven't got to the end yet. Won't be sure.
No, it's good. A lot of people, a lot of scientists who come on the show, a lot of them love sci-fi, so I'm sure a lot of people are going to be happy for the recommendation.
And then the my main wind down at the end of the day activity once the kid's in bed is Baldur's Gate 3.
Ah, yes.
Dual controller, split screen with my wife. That's a perfect way to end the day.
That's beautiful. I love that. I'm currently reading, I don't know if you've ever heard of Becky Chambers. I think I've recommended her books like 5 or 6 times on the show, but I just realized that she had a book come out in 2021 and I just completely missed it. It's called The Galaxy and the Ground Within. I'm on page 25 and it already seems like it's going to be beautiful. I'm really excited. Yeah. I'm also, I love a sci-fi recommendation. Well, thank you very much for joining us. You're welcome back anytime if you ever want to come talk to us more about rats.
Be careful what you say, because
or about zooarchaeology in general, or about your sea changes project. You've got quite a few things going on.
Yeah, I rarely turn down an invitation to talk about rats, so be careful with that one. Thank you.
Well, thank you so much, and have a great day. Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.